Hello, everyone. Welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Finnegan Calabro. Finnegan Calabro completed his graduate training in biomedical engineering at Boston University, where he used computational modeling and neuroimaging to study visual motion perception, including characterizing the mechanisms supporting the perception of motion in depth. He moved to the University of Pittsburgh as a postdoc, where then he became research faculty in psychiatry and biomedical engineering, studying mechanisms of plasticity, both normatively and following the neurologic injury. He now serves as the co-director of the Laboratory of Neurocognitive Development at Pittsburgh and is now contributing investigator on several national consortium projects, including the Adolescent Brain Cognitive Development's ABCD study. His current work involves the use of multimodal neuroimaging approaches to characterize how structural, functional, and neuropsychological maturation contribute to the development of cognitive and motivational behaviors during the transition from adolescence to adulthood. I would like to remind our audience to please address any questions using Q&A box or raise your hands at the end of the talk. Dr. Calabro, thank you very much for coming today, and the stage is yours.
Great. Thank you so much for the introduction and for the invitation to be here today. Like you said, I did my training in Boston, and I was just saying before, Martinos is where I learned to scan, and so it's kind of nice to be back in this forum, even if virtually, and seeing some familiar names here. In addition to that, I just want to say I'm happy to try to take questions as I go if people have any. I know it's a little challenging in this format, but I'm more than happy to answer things as I go. So as Jiang said, I'm Finn Calabro. I work in the Laboratory of Neurocognitive Development at the University of Pittsburgh. And I'm excited to talk about some of the work we've been doing using a multimodal imaging study to understand reward learning through adolescence. Just to set this up, for those of you who don't think about adolescence as a kind of model system, when we talk about adolescence, we think about it broadly as in terms of kind of two main kind of systems that are showing protracted development through this period of life. So on the one hand, we know that cognitive systems are continuing to mature through adolescence, as shown in kind of the solid line here. And this includes aspects of cognitive control and executive function, like working memory, inhibitory control, cognitive flexibility. And certainly all of these functions exist prior to adolescence, but through adolescence, we see a real kind of refinement and stabilization of these processes as we transition into adulthood. And kind of in parallel to that, we know that adolescence is a time of really kind of unique maturation of reward systems. And so we see this in terms of heightened reward responsivity in parallel to maturation, in particularly of kind of subcortical, striatal, and basal ganglia systems. So we've done a lot of work. This is one example of a model kind of characterizing these aspects of development. And we've done a lot of work in conjunction with a former grad student in the lab, Brendan Turnbull Clemens, who's now actually at MGH, to really try to kind of characterize this more kind of specifically. And so he's shown really nicely that we see these kind of aspects of cognitive stabilization across a large number of studies and a large number of tasks. And at the same time, that there are many kind of aspects of reward function that show this kind of peaking in adolescence. And so we're interested in this time of life as where these two systems are really undergoing dynamic flux and how the interaction of these two processes leads to unique adolescent behaviors. So in this talk, I'm going to really mostly focus on the kind of reward processing, although with a bent towards these kind of learning systems that I think kind of bridge these reward and cognitive systems. And just to further kind of preface this, I think when we think about reward processing in adolescence, I think there's maybe a natural inclination a lot of times to kind of focus on the maladaptive aspects of this. So we know, for example, that adolescence is a period of heightened risk-taking and kind of behaviors that might be detrimental to or undermining survival. and can manifest in things like substance abuse and other kind of behavioral kind of phenotypes that we might think of as not advantageous. But I think one of the things we always try to emphasize is that this is, there are two sides of this coin, and that the same kind of heightened reward seeking and sensation seeking is really fundamental to learning that goes on during this period of life when people are kind of out and experiencing the world outside their kind of family environment more. And that how different behaviors translate into rewards is really a fundamental learning process during this age range. So when we talk about these behaviors, of course, there's a long line of work characterizing the neural basis of some of these things, starting with the kind of seminal work of Wolfram Schultz and many others since, characterizing in particular how dopamine is related to these processes. So the work from rodents and other animal systems has shown really nicely that these kind of prediction error signals, so the difference between rewards that you get and rewards you were expecting, seems to be a big driver of dopamine release. And this has led to other models like the temporal difference model that really characterize how we update expectations based on rewards as through a kind of learning process. And at the same time, one of the reasons we're interested in this is that we know that the dopamine system, we know mostly from rodent work, that the dopamine system is also undergoing significant maturational change through adolescence. And so this, what that change looks like depends on what aspect you're looking at in terms of different receptor densities and vesicle quantities and things like that. But it's a period of life where there's a lot of maturational flux in the kind of underlying neurophysiology that supports these kind of reward-related behaviors. So our interest is in really understanding what is changing developmentally through adolescence in the dopamine system to support the emergence of adult levels of kind of reward learning behaviors. And I'll just quickly mention that one of the reasons we're interested in this, of course, is that we also know that simultaneous to all of this, adolescence is a period of life when a number of mental health disorders first emerge. And many of these, including things like major depressive disorder or psychosis spectrum, are disorders that we know implicate the dopamine system. So the data I'm going to talk about today is basically normative, but we're kind of interested in these questions in terms of what the maturation of this system can tell us about the emergence of some of these mental health disorders as well. So I'm going to break the kind of talk up today into kind of three parts of a study that we've kind of recently finished, I guess, and kind of talk about a couple different aspects of the work. So the first part of this work I'm going to talk about is some PET and fMRI imaging we've been doing in the young adult cohort of our population to really characterize how individual differences in dopamine function support specific aspects of reward learning. And then I'll kind of introduce some MR-based acquisitions we've been using to really let us then translate this work into adolescence. So we can't really do PET imaging in our pediatric populations. And so we've been interested in the development of non-invasive MR-based measures that can speak to dopaminergic neurophysiology, and then kind of tie this together and show what these kind of MR-based measures can tell us about the maturation of dopaminergic neurophysiology through adolescence and how that's contributing to the development of reward learning behaviors. So this is really for all the data I'm going to talk about today. This is a study that we conducted here at Pitt. We used, this was all done on a 3T Siemens Biograph scanner. So this is a scanner that lets us acquire both PET and MR data simultaneously. This is kind of the fMRI bit here describes our full sample. So we had about 146 participants that we scanned longitudinally every 18 months, or we tried. So we have a total of about 300 scan sessions completed here. And that spanned the age range of about 12 to 30, which is kind of our window for looking at adolescence and then into young adulthood. For the subjects in the study that were 18 or older at time of enrollment, they also participated in a simultaneous PET study. So this was 75 unique individuals, about 160 visits. And we actually did 2 different PET scans. The first one I'm going to talk about here is a raclopride study to really interrogate striatal dopamine. Raclopride is a D2, D3 antagonist. And I'll come back to that in a little bit. So while subjects were in the scanner, this is kind of what a scan session looked like for us. So we did a 90-minute acquisition for the raclopride tracer. This was a bolus plus infusion design. And what we did is to kind of about 35 to 40 minutes into the scan, subjects performed a task. So simultaneous to the raclopride being administered, we collected a number of structural measures as well as resting state fMRI. And then, like I said, about 35 minutes into the scan, subjects began performing a task. And so the idea here was to try to use this task to really probe the functional and dopaminergic contributions to reward learning. So the task we used was something we developed that is kind of a modification of kind of classical multi-arm bandit tasks. We tried to make it a little more engaging for our younger participants, but still pretty difficult. And I'll come back to why we wanted it to be difficult later. But basically the idea in our task here is that subjects were represented by this little frog avatar, and they were on this 3x3 map, and they had to move around the map and explore where they could find rewards. So they knew that every square on this map had a different probability of giving a reward. They didn't know this, but the rewards were anywhere from 20 to 80%. So nothing was deterministic here. And over the course of about a half an hour of doing this task in the scanner, they had to kind of find where the best locations on this map were so that they could maximize the rewards and therefore, as far as they knew, try to maximize their monetary compensation. So each trial they did about 150 trials of this task. On each trial, we broke it down into 2 epochs. So the first epoch, they saw on each trial, we allowed them the choice of 2 different map locations. So we didn't let them just freely explore the map. We kind of manipulated where they could go. And part of the reason we did this is to match the reward schedule across participants. So everyone would end up getting the same number of rewards, although they didn't know that. So during this first epoch, subjects would see 2 hash marks to tell them which map locations would be available on that trial. And this is the kind of decision-making and reward expectation phase of the task. So they can think about where they want to go and maybe anticipate whether they're likely to get a reward, but they can't actually make a response. After some period of time, those hash marks switch to numbers, and they could indicate with a button box where they wanted to move. And then they would get feedback, and the feedback could be either no reward or 1 or a pile of coins to indicate that they found a reward. So we quantified performance a couple ways here. First, we of course just looked at how often people moved to the higher probability map locations. We also did a post-scan assessment to see how well people recalled where the good places on the map were. But both of those had, well, in particular, the in-scan data has a limitation then, which is we really want to understand not just kind of top-line performance on this task, but what strategies people were using. And one of the problems that we run into here is that strategies like exploration can sometimes be hard to disentangle from just overall accuracy. Are you picking a bad location because you don't know where the good map location is, or because you're exploring the map? So to that end, we also developed a model to try to kind of characterize what strategies people were using here. So we used a reinforcement learning model. For those who aren't familiar, I'll just briefly introduce it. Basically, this is a model that has 2 components. The first is an update rule. So on every trial, you get a reward, and you compare that to what your expectation was for that trial, for that map location. And then you update your internal representation of expected value based on some scaled version of that difference. So this is the prediction error term, and this free parameter here is the learning rate. So how much you update on every trial your internal expected value. The other kind of main part of this model then is a choice rule, which we see down here. And this basically is a way of characterizing how reliably people will choose to move to the higher value square. So even if you kind of know that one square might have a slightly higher reward probability than another, you might not 100% of the time choose to go there. And in this case, the free parameter here that I'll refer to as temperature kind of controls how often you do that. So the analogy here is kind of from thermodynamics, that if you have a high temperature, that's a very kind of stochastic state, that's the red line in this plot, that even if you have a sense that one square is much better than the others, you're only slightly more often than chance going to select it. Whereas if you're at a low temperature, you're in a much more stable state, and you will very reliably choose to move to that location. So we implemented this model. We actually implemented about 50 or 60 different versions of this model and did some model selection to try to see kind of what parameters best describe performance on our task. The kind of best fitting model that we found was actually very similar to what I have shown on this slide. It was a pretty simple model. The only modification we really had to make was that in addition to just having the learning rate, the model seemed to work better if we modeled positive outcomes and negative outcomes as having distinct learning rates. So people might update differently when they had a kind of larger than expected reward compared to a lower than expected reward. So to get a better intuition for these parameters, I'll just try to go through this quickly. We did some model simulations to really understand how these parameters predicted performance on our task. And so this is an example of some of the simulation data to highlight the kind of how both temperature and the learning rate map onto kind of top line performance on this task. So what we can see, first of all, as the color changes here, we're moving from kind of high temperature in blue to low temperature in red. And in our task, at least, that kind of decreasing of temperature is associated with much better performance in terms of the proportion of optimal trials. And this is something I'll just mention is very task dependent, how these will map onto performance. In our task, there were no reversals or anything kind of dynamic through the course of the task. So there was comparatively less value in being overly exploratory because of that. So for our task, at least, these kind of more stable choice rules seem to do much better. On top of that, we had this kind of interesting relationship with learning rate, where we found these kind of quadratic associations. And the intuition here is really that given a kind of limited window of time to do this task, a learning rate that was really low just didn't have time to, that means you're essentially not updating very much on each trial. And there's simply not enough time to actually build a kind of accurate representation of expected value when the learning rates are really slow. Conversely, learning rates that are really high end up having kind of the opposite problem, which is that you end up kind of updating your value representation so much that when you get one unexpected outcome, you kind of forget all the good stuff you've learned up to that point. And so for our test, there was this kind of sweet spot. It turned out to be a learning rate around 0.4 that kind of balanced these things. It learned quickly enough that you know, you'd be able to learn something within the 30 minutes you're doing the task, but not so much that you forget whatever history you've accumulated up to that point. So based on this, we defined an optimality measure of the learning rate. So essentially how close each of our subjects were to using a learning rate right near this kind of peak point. So when moving into kind of actual data, then when we look at fitting this model to all of our actual participants, the first thing we did is just say, how well does this model actually fit our observed data? And what we found is that we ended up pretty close to a median split in terms of subjects that were clearly well fit by this model compared to subjects that were not necessarily better fit by this model compared to a guessing model. And that median split, I think, reflects partly that we made this task pretty hard. There's 9 map locations to learn probabilities from over the course of the session, which is a lot to keep track of and a lot to learn in a relatively small number of samples. So our population was kind of split half and half between people that seemed to figure this out pretty well and people that didn't seem to. If we look at the kind of raw performance measures split between these two groups of people that were and were not well fit by the reinforcement learning model, we kind of, as expected, see that if you look at how often people made movements to the optimal map location, the learner group did this pretty well. They pretty quickly were able to figure out where the good map locations were. Whereas the group that wasn't well fit by the model, they were above chance statistically, but only slightly. And they never really seemed to figure out kind of the structure of the map. We also see this in the post-scan assessment. The learner group recalled much more about the kind of structure of the map after the scan. But importantly, we didn't see any differences in response times between these two groups. So that it's not like the non-learners were falling asleep or not responding or not engaged. They seemed to be doing all of that. They just weren't learning very effectively. So our kind of first big question here was, given this group difference in performance between these, what we call learners and non-learners, what is the difference, first of all, in the dopamine system between these groups? So to do this, we looked at the Raclopride data, and I'll just kind of introduce this a little, especially for those who aren't familiar with PET. Basically, this is an example of time activity curves that we acquired, like I said, using a bolus post-infusion protocol with our Raclopride tracer. And basically, this is showing time activity curves for 3 striatal regions, as well as our cerebellar reference region. You can see the cerebellar region we're using in lieu of blood draws. This is a region that we define because it has very low dopamine receptors, so it provides kind of a good reference for what's happening in blood. I'm not going to go into the modeling approach here in huge detail, although I'm happy to talk about it a little after, except to say that in order to then kind of translate these time activity curves into something physiological, we used we started with a simplified reference tissue model. So this is a multi-compartment model that characterizes how the tracer is moving between blood and tissue, both in regions of interest, which for us was voxels within the striatum, as well as in our cerebellar reference region. We made one modification to this that had to do with the fact that we're doing a task during this scan. And so what we did is, instead of modeling, sorry, I should have said, the kind of goal of this model is to kind of develop a system of differential equations that allows us to assess the binding potential. So we can take this whole time activity curve and collapse it down into this binding potential number that tells us what's the receptor density in that region of interest. Because we were doing a task, what we were really interested in was not just the kind of binding potential, but what our task was doing to them. So we modified this SRTM model to allow us to try to estimate the effect of doing the task on the binding potential as measured by Raclopride. So the intuition here is that pre-task you have some amount of dopamine floating around endogenously, but some number of unbound receptors. And the binding potential is going to give you a measure of the kind of density of these available dopamine receptors. When we do a task as a type of challenge, what we think or hypothesize is that there will be actually dopamine release in this region of interest that causes an increase in the amount of dopamine and a decrease in the number of unbound dopamine receptors. So when we do the task, we expect the binding potential might drop during the task in response to kind of the reward aspects of this task. So we modified this task. The important part here, we modified this SRTM model. The important part here is that we changed how we characterize the binding potential to instead be this kind of step function change during doing the task. And this is based on some really nice modeling work that has developed over time from a number of groups. Evan Morris at Yale in particular helped us a lot with developing this approach, our approach for doing this. And you can kind of see an example on the right here for one region of interest. The green line is the model fit to this data without considering task effects, whereas the black line is the model fit when we include this kind of task-related change in binding potential. So, what that gives us then is essentially 2 measures of dopamine function. One is the kind of baseline binding potential, which is kind of a trait level measure of receptor density. And then the task effect, which is how much the binding potential changed during the task. So if we bring this back to our behavioral data and kind of look at those groups, the learner group versus the non-learner group, the first thing we saw is that if we look at this with baseline binding potential, there's no difference between the two. So this is a map of baseline binding potential across the striatum, and you see a nice gradient with slightly lower binding potentials in the ventral striatum, increasing in the caudate and putamen. This is consistent with a lot of prior work. But importantly, if we looked at this by region or voxel-wise, nowhere that we looked was there any difference between our learner and non-learner groups. So just simply having more or fewer, available dopamine receptors didn't seem to predict whether you were going to learn on this task. On the other hand, when we look at the task effect, what we found, if we look, first of all, just across the full sample, we found two pairs of clusters that showed significant task-related change. So we found a bilateral ventral striatal region and a bilateral region of the putamen that both indicated a significant reduction in binding potential when doing the task. When we mapped this then onto our learner groups, we found that the putamen didn't differentiate at all, which might support that it's doing some kind of motor or other kind of function. But the ventral striatal region, which is our kind of, we know as a big reward-related region, showed greater dopamine response among our learner group compared to our non-learner group. Excuse me. So this is kind of a nice finding for us and kind of suggests that the learner group here is eliciting greater dopamine responses, even though, like I mentioned before, they're not actually getting more rewards. Everyone is getting the same amount of rewards. The difference between these groups is really in the extent to which they're able to learn from those rewards. So we wanted to unpack this a little more. And so we looked at this data in conjunction with our reinforcement learning model, and in particular with the different parameters that emerged from that model. And what we found is that there was no relationship between dopamine release and temperature, the kind of choice parameter here, nor with kind of the learning rates people used following negative outcomes. But we found a significant quadratic relationship between the positive outcome learning rate and dopamine release, such that subjects that used kind of intermediate values of learning rate were the ones that tended to have the greatest dopamine release or response. And this is reminiscent, I think, of the simulation data I showed at the beginning, which suggests that these intermediate values of learning rate are also the kind of task optimal learning rates. So if we transform this data instead of plotting against learning rate and just plot against the optimality of the learning rate, we see again this significant association that the kind of more optimal the learning rate people were using, the greater the dopamine response seemed to be. So I think both of these results kind of argue then that the dopamine response we're seeing in this task is really a fundamental component of the learning people are using, and importantly, seems to really be about kind of the learning strategy. It's about the choice of the learning rate more than the outcomes themselves. But there are some limitations here. For example, our measures of dopamine are very slow, right? So we did a 90-minute scan, and from that we got two measures. We got one number that told us the baseline binding potential, and one that's kind of an index of kind of aggregate dopamine response over the entire task session, the entire 30 minutes of doing the task. And so we don't really know when within that session this dopamine was coming from, and we'd really like to, characterize the mechanism that is leading to dopamine response. So to kind of interrogate that, we looked at our fMRI data while subjects do the task. And so I mentioned before that there were kind of two main epochs of this task. There was the expectation phase of the task, and then the reward receipt or outcome phase of the task. This I'm just showing kind of main effects of activation at each epoch. These look a lot like kind of canonical reward networks that are kind of frequently associated with these aspects of reward function. But what we're really interested in then is saying what of this activation scales with the dopamine release that we see. Can we correlate on a subject to subject basis where bold activity seems to correlate with or scale with dopamine release? So when we did that, the kind of interesting thing to us that emerged is that we were able to find significant clusters of bold activation that were correlated to dopamine release, but these were exclusively occurred during the expectation phase of the task. So kind of surprisingly to us initially, we didn't see any correlations between bold reward responses and the dopamine response that we measured with PET. Everything we found was during the expectation phase. We found a number of regions, some prefrontal regions, as well as the ACC, and then some thalamic activation as well. And in all of these regions, I'm showing the kind of correlations for a couple of them as examples here, but in all of them, the relationship we found was greater bold reward expectation activation associated with greater dopamine response. So I think this kind of builds on the PET data to kind of, again, suggest that this response that we're seeing, in particular with the PET dopamine signal, is not really just about signaling the presence of a reward, but really as a kind of fundamental substrate to learning and the formation of expectations. So, I mentioned at the beginning, our research is really about adolescence, and we're really interested in translating this work into adolescence. And a major limitation there is that we can't do PET imaging in pediatric populations, at least not easily. And so we've been interested in how do we then leverage these findings to understand what's happening developmentally. So I'll just briefly introduce one approach that we've been taking to look at tissue iron as a potential non-invasive marker of dopamine physiology. And the rationale here comes from the fact that ferritin in tissue, so as opposed to kind of blood iron levels, is a kind of byproduct of neurotransmitter synthesis and is in particular a cofactor for the production of dopamine. And so there's been this kind of line of thought that maybe measuring ferritin, which of course is ferromagnetic and therefore a good candidate for kind of MR-based acquisitions, might provide an opportunity to look at something correlated to dopamine physiology non-invasively. So there was some initial really nice work done to kind of validate this idea at the microscopic scale that if you look really at kind of low levels at individual neurons, dopamine tends to co-localize with iron really nicely. So the places where you see iron show up tend to be also the places of high dopamine content. And we knew for a while that from some clinical work that if you looked at neurological disorders like Parkinson's, as well as resting leg syndrome and ADHD, that there were differences in brain iron typically associated with these conditions. And we know that these are conditions that implicate the dopamine system. So there's some kind of circumstantial evidence that supporting this idea that maybe tissue iron is telling us something about dopamine physiology. So one of the things we did as part of this study when people were in the scanner collecting structural data is to acquire tissue iron specific scans. So you can measure tissue iron really nicely using T2 star and T2 weighted imaging because iron content changes the time constant decay rate of the T2 star signal. So this is an example across different echo times of a low versus high iron voxel, and just kind of schematically showing that as iron content increases, the decay rate of the T2 star signal scales proportionally with the iron content. So we used a scan that was a multi-echo acquisition, so that I think we actually acquired 5 echo times to try to characterize the time constant here. So what is the decay rate of the T2 star signal? And by comparing this to the decay rate of the T2 weighted, the T2 signal, we can kind of solve this for a quantity called R2 prime, which is the inverse of the T2 prime signal, which is derived again from the decay rates of the T2 star and T2 images. So when we compute this, then we can get a map that looks like this. This is now going to be a measure that should be proportional to iron. And you can see, for example, in the pallidum, which we know is kind of the region of the brain with the highest iron content, the R2 prime signal is really hyper intense, but that we also have kind of measurable signal throughout the striatum to give us a kind of quantitative measurement of tissue iron. So one of the things we did with this then is to look at how this mapped onto our PET data. I've talked about the raclopride data. We also collected DTBZ, which is another PET tracer that targets the dopamine system, in this case, presynaptic vesicles. And what we found is that if we looked at a correlation between R2 prime, so our measure of tissue iron, and in particular, DTBZ, we had a positive correlation such that higher iron content was present in subjects that had higher binding potential of this kind of presynaptic dopamine measure. This was even more pronounced if you control for age, because tissue iron tends to accumulate with age. And if you control for individual variability by looking at longitudinal change, this is over the course of 18 months, we again saw a significant relationship. So, we think that this is providing some evidence, on top of the molecular and clinical work that's been done, that both cross-sectionally and longitudinally, individual differences in tissue iron seem to be telling us something about individual differences in dopamine neurophysiology. And I'll just kind of quickly mention, we've done a bunch of work looking at this. Ashley Parr, who's another faculty member in the lab, has done a lot of nice work characterizing this for anyone who's interested, looking at how these tissue iron levels vary across regions of the striatum, as well as how they change developmentally. And also looking at how this maps onto functional consequences, neural functional consequences, and showing that individual differences in iron are significantly related to the strength of frontostriatal connectivity, which we see both cross-sectionally and longitudinally. So we think this is kind of building a story where we can use tissue iron then hopefully in developmental populations to tell us something about how these dopaminergic systems are maturing. So that's the kind of last part I want to talk about, is whether we can leverage this tissue iron data to understand the neural basis of kind of development of reward learning strategies through adolescence. So to kind of revisit and jump back to the first part of this talk, we look then, now this is our full sample, so this is ages 12 to 30. If we look at performance on our task, the first thing you'll definitely notice is that there's a huge amount of individual variability here. Some people are just really good at this task and a lot of people are just really bad at it. But on top of all that individual variability are some highly significant developmental changes. So people are getting better at doing the task. If we look at in-scanner performance, as well as if we look at post-scan recall performance, they're also getting faster at the task. So to kind of understand what's happening developmentally, the first thing we wanted to do was to revisit that reinforcement learning model and say, can we understand computationally what's allowing people to get better at this task? So if we look at this data, and we did, the kind of first thing that jumped out was that we didn't see any univariate relationships between the parameters of our reinforcement learning model and age. So looking at, these are kind of three main free parameters, the positive and negative learning rates, as well as the temperature parameter, how stochastic they were, as well as the transformations of this data to characterize how optimal the learning rates were. None of these had univariate relationships with age. So, we were kind of left wondering, well, what is it that's changing that allows people to get better at this task? So when we investigated this data a little more, one of the things we noticed is that these parameters, and people that have done this kind of work probably have experienced the same thing, these parameters are really highly correlated with each other. They're not really independent assessments of performance or computational strategy. Instead, what makes someone good at the task is not just having, say, a higher learning rate or a lower temperature, but kind of where these parameters fall relative to, in terms of the relative rate of these two things. Excuse me for 1 second. Sorry. So what we did then is to say, well, instead of looking at these parameters individually, let's try to combine them and look for patterns across the different parameter space. The way we did that was to take these kind of three main free parameters and two derived parameters and run a principal component analysis. So if we ran this with these 5 input variables, we of course get 5 principal components out. One of these really jumped out. So I'm going to focus just on this third principal component and that emerged. And the reason this one jumped out is that if we looked at the kind of in-scan and post-scan performance, the expression of this component was a significant predictor of how well people did on the task. And interestingly for us, not only could it explain individual differences in performance and recall, it also showed strong developmental change. So the expression of this component increased significantly with age through our sample. If we try to unpack a little what's going on in this composite parameter, we found something pretty interesting. So a positive value here was associated with, I should go back to here, we can see the legend, positive values of the, in particular, the positive learning rate optimality. So using an optimal learning rate was associated with greater expression of this composite parameter, as was suppression of the temperature parameter. So the people that had kind of high component 3 values here are the people that both used highly optimal learning rates and low temperatures, meaning that they were not stochastic. They were very exploitative in what they were doing. So we can kind of then try to map these concepts onto the different ends of the axis here. And one of the things we've kind of tried to characterize with this is that this might be catching a shift, whereas at the younger end of our age range, people are using high temperatures and low optimal learning rates, consistent with more exploratory strategies. So they're more stochastic in their choices. And I'll just mention that, importantly, this is undirected exploration. So this is kind of the stochastic choice exploration that people seem to be using. We tried a number of versions of this model where we would explicitly model directed exploration. So do people seek out map locations that, for example, they haven't visited many times? And we couldn't in our test find support for that. But we do see that there's this kind of increased use of these kind of stochastic exploration strategies at younger ages that then shifts as subjects age to kind of less stochastic decision-making, but more optimal learning rates. And not only did this increase with age, this significantly mediated the age-related changes in performance that we saw. So the kind of shift from negative to positive in the expression of this composite parameter seems to be a driving factor, or at least an explanatory factor for how people are improving on this task through adolescence. So, of course, we're interested then what the brain basis of this is. And so we looked at this in conjunction with our tissue iron data. So this is an example of a tissue iron map from a 12-year-old versus a 30-year-old. And first of all, you can see that tissue iron is accumulating pretty strongly. You can see in the pallidum, these regions getting more intensely bright. Also in the ventral striatum, the level of iron is increasing across our age range here. And what we found, so I mentioned on the previous slide that there's this shift in this composite parameter from exploration to optimal learning. What we found when we looked at the tissue iron is if we looked particularly in the ventral striatum, which is the region that we found with our PET data implicating dopamine in this task, there was a significant relationship with iron content. So subjects that had higher levels of ventral striatal iron were also the ones that tended to use higher values of this composite parameter, reflecting the use of optimal learning strategies, whereas the kind of low iron subjects tended to use more exploratory strategies. And this was true whether or not we controlled for age. It's important to control for age here so that we're not just reproducing our age effects. Even controlling for age, we see this association to kind of characterize individual differences on this task. And this also mediates the age-related change. So age-related changes in tissue iron content were significant mediators of the shift in this composite parameter from exploratory to optimal learning. So we think this builds upon what we saw in the PET data, implicating, in that case, dopamine itself in the ventral striatum as supporting reward learning, and now using these kind of more static kind of structural measures from tissue iron that there again seems to be a mapping between this, what we think is this useful index of dopaminergic neurophysiology, and that as it matures, that seems to happen in parallel with this kind of strategy shift from exploration to kind of effective and optimal learning. The last thing I'll mention then, of course, is that, I mentioned this tissue iron seems to be a pretty structural, stable measure. Again, like with the PET, we're interested in what the kind of functional substrate is for this. So we again turn to our fMRI data And in this case now said where across our full sample of adolescents and young adults, where is this composite parameter represented in the brain? And so if we look at that voxel-wise, again, we looked at both the expectation and reward outcome phases of the task. This is in particular clusters that we found where bold activation scaled with the reinforcement learning composite parameter that I've been talking about. It was particularly during reward outcome in this case. And in particular, the associations that we found were age-related interactions. So an interaction between age and the expression of the composite parameter predicting bold data. So to kind of unpack what these interactions are doing, I should say, first of all, we found 3 significant clusters that showed these interactions. One was this kind of ventral medial prefrontal region, which we know is a strong decision-making area, as well as bilateral posterior parietal region. So to kind of unpack these, we looked at what the bold signal was by age and by composite parameter. And what we found for all 3 of these regions was a similar pattern where if we looked at our adolescent population, so our under 18 population, there was a negative association. So what this means is the highest bold activation among adolescents was observed among those who had the lowest value of this composite parameter. So again, a low value of the composite parameter is we think associated with more exploratory strategies. So at least during adolescence, activation of these regions seems to scale with use of these kind of exploratory strategies. Whereas by the time you get into young adulthood, that has kind of flipped. And now the greatest bold response seems to be present among individuals that are using less exploratory strategies and more optimal learning strategies. So we think that this kind of, builds on the PET data in a kind of nice way to kind of really emphasize the role of dopamine in learning, at least on this kind of task, and the contribution that dopamine has to the maturation and the development of these reward learning strategies. And I'll wrap up there and just kind of reiterate that, I think that this is really speaking to the role of dopamine, not just in signaling rewards, but in really dictating reward-related learning heuristics and strategies that seem to be strongly linked to ventral striatal dopamine, both from our PET data and from our tissue iron data, but also importantly supported by a network of cortical regions that are interacting to kind of lead to these kind of behaviors developmentally and the emergence of adult levels of reward learning behaviors. So I'll kind of wrap up there. I just want to acknowledge we have a great group here at Pitt working on this, in particular, Bea Luna, who's the head of our group, and Ashley Parr, who's another faculty member of our group, and a large number of collaborators, some of whom he's listed here. And I'll, in particular, single out Julie, who I think I saw on the call before and is now at MGH and was really fundamental to us kind of figuring out how to set this study up and teach us about PET data. So thanks again for having me, and I'm happy to answer questions.